So this optional office hour is to do a little walkthrough of the lab report and answer any questions you might have as you're getting set to do this assignment. Before we get started, are there any burning questions you want to address or discuss right now? Maybe we can get out of the way. Go ahead and unmute or raise your hand if you have a question that you'd like to have addressed kind of up front, especially if maybe you can't stay here the whole time and you're worried your question won't be answered before you leave.
Yeah. Hi.
Hi there. Go ahead.
I just want to make sure if I understood correctly. The reiterating the elimination process. So we make the flow chart, and then after that, we're gonna state again the whole elimination process, like kind of short way, in a short way. Is that correct?
Yeah, so the flow chart's going to be complete in the sense that in the flow chart, you must name every species.
All those species, right?
As they were eliminated. So for instance, if you're in the Enterobacteriaceae and your first test is the indole test, you name every species that are eliminated by the indole test and make sure that, you know, that species is now gone. It should not appear later in your flow chart.
Right.
In the body of the discussion, you walk through that flow chart in complete grammatically correct English sentences, you know, like not a bullet point or anything. It's the unknown number 41. The first paragraph of that would be how you got to your group ID, which does not have to be on the flow chart.
Okay, including that part. We have to include that part too.
Yeah, so the first paragraph of your discussion would be unknown number 41 was gram positive, let's say, and rod shaped. The test for catalase was positive, indicating that this organism has the enzyme catalase and can detoxify hydrogen peroxide. Subsequently, the endospore test was positive, indicating that this unknown belongs to the group Bacillus. That's your first paragraph. The next one starts to walk through the actual flow chart. So the first test run for the Bacillus might have been, let's say, I don't know, motility. The first test run for this unknown was motility. The motility test was negative or positive, indicating whatever that indicates, does or does not have flagella, does or does not have motility. This test eliminated 27 species.
Okay.
Right. So at that point, you still say how many species were eliminated, but you don't list the names of the species over again. You just say eliminated 27 species.
That's the number.
Say, what test was done? What was the result? Just positive or negative. You don't state whether, you know, what the tube looked like, because that's already in your results. And then you say what it means biologically or structurally, and then you say how many it eliminated. And then you walk through in chronological order until you get to the last one.
Wait a second, wait. That part, I was confused because in the instruction section, we have another section that we discussed the structure and the metabolic, the test. Do we have to combine together? No,
are you talking about in the discussion still?
Yeah.
Okay, let me do a screen share and we can look at the instructions together, and then I can help to clear up any confusion. I hope that you saw my announcement. There was another set of instructions accidentally included that was not the same, exactly the same. It was very similar. But if you go to the Unknown ID project, the instructions you should see are the ones that are called instructions, Unknown ID lab report instructions. Is it this page that you're talking about?
Yeah.
Yeah. Okay, so let's go there.
So there is a part where we have to discuss the structure and metabolic features.
Yes. So right here is what you're talking about.
Yeah, exactly. Yes.
Yeah.
Do we combine?
Yes.
Do we combine this and the elimination process?
Yes.
Okay.
Yes. I'm sorry that wasn't clear. I can write that more clearly. Also, I need to delete this step 4 because you don't have to do step 4. It's optional, right?
Yeah.
So you just do the extra credit part if you want to do step 4, and that's a separate Dropbox altogether. Yes, so good question. And it doesn't seem like it was very clear here. And to be honest, looking at this, so once you've done the flow chart, if all you do is this part and you don't walk through your eliminations, I might just be fine with that because it is so unclear and some people may have started writing this.
It's okay. I've already wrote it, but you know, I can incorporate it. I mean, no big deal.
Yeah, no, but you're not going to be the only one who's confused by this. The other thing that I'm thinking about is that this does not make it clear that you have to write about the group ID, which you do, but that one is important. So I will make a note to change that and announce it so everybody knows. Yeah, gosh, I'm sorry. I didn't realize that this was so poorly written. I don't know what happened when I wrote it. Okay, so yes, I'll amend this and I'll announce it. But I'm not going to require something substantially different from this, except for that first paragraph has to talk about the group ID. That's really important. Yeah. Does that clarify for you?
Yes.
What to do? Okay.
Yeah,
so yeah, I will accept if the eliminations are spelled out here, then and all you want to do is just walk through what each of the tests revealed structurally and metabolically without saying what the eliminations are over again, I would accept that. Yeah, that's also fine. Okay, let me just check my little chat ski here. And also, Roman says, if I already put step 4 in the report, should I delete it? No, that's fine, Roman, don't worry about it. I just won't grade that part. But if you have it in there, that's wonderful. You know, it's a nice feature to have. It just won't make a difference to your grade. And I don't want to make you, you know, go back and have to take work out that you've done if it's not something that's like against the rules, actually, if that makes sense. So leave it in. If you've already done step 4, leave it in. That's completely fine. The things you want to be careful about are the things that you can't do, like you don't want to restate your observations in the discussion. Okay. Let's see. I want to make sure also, let me stop share for a second. Yes, there's nobody in the waiting room. Okay. So before I roll forward and sort of my little thing, that was a great question, Julia. Thank you for asking it. Are there any other questions like that where you just kind of want to ask them up front, just so you make sure you don't miss out on the answer that you've been searching for? Jade, go ahead.
I just have kind of a silly, quick question, but is it okay if we use the example title that you gave us as our title, or do we need to come up with our own?
Let's see, what was my example title? I don't even remember. Oh yeah, I see it. Identification. Yeah, identification of an unknown is fine. I'm fine with that. Yeah.
Okay, I wasn't sure if we were supposed to be creative and come up with our own, or if we could just use their example.
Yeah, it's fine if it's boring. That's okay.
Okay.
Great question. Here's the one I don't want to see. I don't want to see lab report. That'll just make me sad. I mean, you guys are past that. Okay, Jessica, go ahead. What's your question?
Is there anything specific in the resource part that you want? Because I'm having kind of a difficult time with that part.
Oh, the research part, you mean? Or
yeah,
like you know, the 3/4 of a page where you talk about the species? Is that what you're asking? Yeah. So it's important to mention anything that's sort of especially relevant to people. Like if this is a human pathogen and you don't say that it's a human pathogen, I'm going to wonder if you did any work, right? So if it is a human pathogen, you want to say something, and most of these will have some kind of, and what do we know about that? Like what kind of infections is it normally associated with? And there are a few that are just environmental, and if they never cause infections, you can say that. But you do want to make sure that if there's something particularly important, like it's famous for being a bioterror agent, or it's, you know, well known to cause UTIs or something like that, that should definitely appear there and sort of, you know, should be referenced. And then also you can talk about what people know about it in general, like what are its its features. And for that, you could go to the Burgies. You can just crib the Burgies if you want. The Burgies will have, there's not just the tables that you guys have been working from for your eliminations, but there's an additional section. Let me show you what I'm talking about. And probably some of you have seen this already. There is another page where, let's see. the whole group is described, and in it you can usually find some fun tidbits about your particular organism. So let me just share my screen. Okay, we are there. That's not, I think this is what I want. There we go. Let's go to the Unknown ID project module. Where is it? Oh, here it is. Sorry, the Burgies module. So you'll notice that there's a second thing here. There's the tables, and then there's this document that says Bacillus pages, and it gives the page numbers from the Burgies. This document will have a lot of information in general about the group, which you're allowed to talk about. You know, don't take a whole bunch of time, but you can say that the Bacillus group in general is known for these things. You know, some of the features that you're going to mention would be things you already know because you had to use those tests in order to identify this particular species. This link is not working. Let me just find another one. But also you can find your particular species. Let's see if I can get this to work. Canvas has been really weird today, so I don't know what's going on. Maybe I can find it on my desktop. Hold on one second, guys. Uh-oh, there we go. Okay, this is just not coming up at all. In any case, if you click this link, I swear to you that there are a whole bunch of pages that talk about the group right at the up front that say, you know, generally this group is, let's say, fluorescent or catalase positive or whatever it might be. And it talks a little bit about that. So you can paraphrase that information and cite Burgies. And then if you continue into the document later, you'll find your actual species on a little paragraph blurb about it that says this species is known for blah, blah, blah. And it's all like biological and metabolic test data. And so you can also talk about that. And then just kind of do a look around and see what people know about this organism, what kind of things that it's normally are described as. So does that help at all? There's not just one right answer, but I do want to see that you kind of made an effort to look around and see what people know about it that's important.
It does. And so when we do the in-text citation, do you want us to just write like Burgie and then 2000? Yeah.
So the citation that you need, Canvas is having a little bit of a meltdown right now. The citation that you need is in the Burgies module. Let me find it and show you where it is. Not that one. Shoot. It should be right at the start of the module. There was a little announcement about this a few days ago.
Yeah, so I put that under references, but I saw you wanted us to have like in-text.
Oh, yes, I'm sorry. I misheard you, and you were very clear, and I'm just a bit of a scatterbrain. Yes, Burgies et al. 2000 would be perfect.
Okay.
Yeah, for the in-text. So the citations at the end, the list with the full citation will have this. But if you say something from that, you read the little blurbs that I just talked about, what the group is like, what your species is like, and then you cite it in the brackets, it would say Burgies et al. 2000. Exactly right.
Okay, perfect.
Yeah. Great question. Anyone else have questions or comments or concerns right out the gate? Jessica. Oh, other Jessica had her hand up as well, but no.
Oh, that was me. Sorry.
Go ahead.
So I have a question about my species. So it is Salmonella enterica subspecies enterica.
Okay.
I'm looking at the Burgies manual that you tried to pull up on Canvas. It worked for me. And I'm looking at, I see, like the subspecies, typhi, paratyphi, all this stuff. And then when I did the ID gene sequence, it came up to be, the Salmonella enterica subspecies Enterica, but it also came up to be like the Sarovar, like Typhi LT2.
Yeah.
So am I researching just the species that I found that it said the Salmonella enterica subspecies Enterica, or okay, that's it,
Yeah.
right?
Yeah. And if you want to talk, like if you find out some cool things about its brother, you know, and you want to talk about that, you can just make sure it's not the bulk of the section, that you focus mostly on your species. But if there's something
yeah,
you
'cause
run into and you say, hey, this close relative does this cool thing or is often confused for this or whatever, that's absolutely fine.
Okay, yeah, because I put that in my research. Like I put like in widely developed countries, there's non-typhoidal ceravars of S. enterica that are important causes of diarrhea.
Yeah,
So like I just put, yeah, because it says there's like different kinds of it. So
Yeah.
I just like put little blurbs in there, but not, it's not my full, you know,
And
research.
to be honest, like you are only required to go to the species level in this. And so, you know, if you have research on Salmonella enterica, other substrains, other ceravars, and you want to include those, that's not a violation at all.
Okay, perfect.
Completely within the purview of what you need to do.
Perfect, thank you.
Awesome. Jade, go ahead.
So I have two questions. The first one is, do we need to submit our initial journal along with the lab report, or does the check 3 kind of count as that submission for the journal?
Great question. So you need to submit the final updated version of your journal to the lab report Dropbox. So I know that that's the one you want me to grade at the end of the day. Even if it's identical to the one in the check 3 Dropbox, you don't want me not being sure or forgetting that you said that, or like, you know, so it to be completely unambiguous, and also so that I don't have to go between Dropboxes and lose my train of thought, which is going to happen for other reasons. Put everything in that lab report Dropbox, including your updated lab notebook, even if it's identical to the check 3 notebook that you've already submitted. Yes. Great
Okay,
question.
so would it be okay if I just attached it to the lab report document? I'm not sure how to, usually after you submit something, it's like, okay, you've submitted one thing. I'm not sure how to submit two things at the same time.
You just submitted another thing, and it will
Oh,
look
okay.
weird to you, but I will see it. If I notice
Okay.
anything weird, I'll tell you, but I would not attach it, so that I'm clear, that you're clear, that you know the difference between these two documents. And attach, and don't, yeah, submit them separately. And if it's not letting you submit them both at the same time, don't worry about it. Just submit one and then submit the other. I can see them both. There's a little drop down menu where I see everything that you put into a Dropbox ever since the beginning of time. And, you know, once in a blue moon, if something doesn't show up there, I will email the student and say, hey, I don't see this here. What's going on? And, you know, typically there's no problem. They can just get it to me, but that's extremely rare. So it should be fine. If you're worried about it when you submit it and you're like, I'm just not gonna be able to sleep tonight, unless I know that it's there, you can email me and ask me, do I see both things? And then I can just say, yeah, I see them.
Okay, thank you. My next question is it okay if we gather all of our information from online and not use the Bergies pages?
Yeah.
Is that okay?
Absolutely. You decide what sources you want to use, as long as they're legit, peer reviewed. The Bergies is a handy source. It's got a lot of fun info, but it's not really like about how these, usually about how these bacteria are relevant to people, and it's not topical or updated. So it's more, you know, what is the biology of this organism? And if you've got other sources that talk about the things you're most interested in and that are relevant to people, then go to town. I don't care.
Okay, thank you.
And with that said, of course, it matters what the references are, but I think you know that, and I'll cover that a little bit later.
Yeah, I trust me, I didn't use any wikis or anything like that. I tried to find .govs and all that, so.
.orgs. Awesome. Okay, any other questions? Super stellar questions so far. Any other questions? Anything else on your mind, guys, before we just do my little walkthrough? Roman, go ahead.
Hi, so what I on the instructions, it says like when you do your references, you put the author's name, put ETAL if there's more, and then you put the date. What I've noticed in a lot of the like official, like CDC and the health.gov, a lot of them actually don't have an author listed. So would it be okay just to list the website and the dates when the article was revised or written?
The best thing to do, the convention in that case, is to do an abbreviation with the date that is similar enough and unique enough that I can find it in your reference list. So for example, let's suppose that you pulled information off of one of the CDC websites, then, and that's in your list, like the long URL is in your list. Then, and if it's the only CDC source that you use, you didn't use like 10 CDC pages, then you could just write CDC 2000 or whatever, or even just CDC if there's no particular date that's easy to find. And then I'll know that's the CDC link or the CDC reference in your reference list. If there's multiple CDCs, then you have to do something more distinctive. Like maybe one of the CDC web pages had, you know, pathogenic species of Sierra Leone or whatever. And the other CDC website was what to do in coronavirus. And so you would have CDC with a little bit of extra text that would help me identify uniquely which reference you were referring to. Does that help?
Yes.
So don't
and
include the whole website, the whole URL, because that just makes your text unnecessarily difficult for me to scan. So do like instead like CDC with some unique identifier if that's necessary.
uh-huh. Okay. And can I also ask one more question?
Yes.
So you did say, so like when we write our lab report, the official one, not to like repeat our observations,
Yes.
correct? For me, it finds it a little difficult. For example, I've tried to explain that this test made the bacteria, I don't know, obligate anaerobe.
But that's not an observation. That is an interpretation. By observations, what I'm talking about is blue or cloudy or yellow, or there was a gas bubble, right? So the observations are the things that you write down that you see with your eyeballs and that help you figure out what the test is telling you. In the discussion, what should appear is a motility test was positive, indicating this organism has flagella and is motile. Right? The thioglycolate test revealed that the organism is an obligate aerobe, suggesting that it's only capable of doing aerobic respiration for survival, you know, that kind of stuff. So it's not difficult to do it. And it can be a habit that you just kind of need to break if you're used to explaining everything very thoroughly, but it's kind of this artificial separation that we do in science. The observations are already done. They're in the results table. So for the discussion, you would say this test was negative or positive, and that tells you whatever it tells you. And just don't go back and refer to the visual data. The only exception to this is like when you're describing the cells, you can say that they were rod or round because that's relevant to kind of walking through that dichotomous key. But don't tell me that they were pink. You would just say they're gram negative. Does that make sense?
Yes. Okay.
Yeah. And just to add to what I know, I'm pretty sure that you understand this, but just to make sure for people who are listening later to the question about references, Roman was asking about in-text citations, what to write. Like when you say something in your discussion, the body of the text of the discussion where you're writing your paragraphs, and you use information from a source. At the end of that sentence, there is an in-text citation in brackets that's a little abbreviated indicator of what your source was. And then later on, something like CDC 2000 or Burgess et al. 2000. Later on, there's a more complete reference list that lists all of those in-text citations, but the full source so that I can find it. So that's where what Roman was asking about. That's where he would put the full URL if it was a website. But in the in-text citation, it would be a smaller little thing that tells me which link to look at or which reference to look at. Okay, any other questions? Anything that's burning a hole in your mind? Let me look in the chat and just see if I'm missing anything. And I see, okay, perfect. Awesome. Okay, well, I was gonna just run through these instructions and let's see, there it is, and kind of make sure that everything was clear. What in the hell happened to my sharing screen? Yikes. Hold on 1 second. Uh-oh. Canvas is really mad at me right now. Give me 1 minute to just get this sorted. It's just super slow. Oh dear. Okay, here we go. All right. Perfect. So I just want to walk through this and kind of reiterate some of the important things and kind of point out the places where people sometimes make a mistake. So I just want to be clear, the title page is a separate page. Okay, all that's on it is your title. And as Jade mentioned, you could steal my title if you want. You don't have to, but it should be something like this. This is a very active scientific sentence. It tells you what was done. There was an identification, and what happened was that it was an unknown bacteria that was identified. So please don't say lab report. Make sure that the title page has your name and the ID number of your unknown. This is very important. The results section. So this is where this differs from the last instructions that I had here by accident. I'm not asking you to write an introduction. There's a results section. The results section only has a table. There's no need for a paragraph. That paragraph is descriptive based on stuff you would have been doing in in-person lab. So this is just a results table. And what would that look like? It's basically 3 or 4 columns. The first column is the test, right? What's the test called? And this should be every test you did, including the gram stain, including catalase or oxidase or thioglycolate or whatever your early group ID tests were. Every single test should appear on this table that produced data. So things like, let's suppose you ordered the propionate test and the technician said, we don't have that test. You don't include that here. But if you ordered the motility test and it came back inconclusive, then you would actually include that here. The date, right? The date is when you received it. So that's when you actually got it. So I can find that if I need to look that up. Your observations are really important and they should be complete. So what was the color, if that's relevant? So don't just describe everything for the sake of description. What are the relevant descriptors of this test? If it's a motility test, the relevant descriptor would be that there was growth throughout the tube or just growth in a single skinny line. If it's a growth test like pH or temperature, the relevant observation is if it's cloudy or clear, indicating there's growth or no growth. If it's an MSA plate, the color and the growth is important. If it's a fermentation test, the color of the broth and whether or not there's gas in the Durham tube is important. So whatever those important descriptors are, you include them here. And then if you want there have a final column, you could have positive or negative, but don't put anything in here like this says that the organism is, has the enzyme catalase, or this makes this organism an anaerobe, or, you know, these are all interpretations. So observations only, and if you really have to have some kind of extra thing in there, positive or negative is accepted. Okay, the discussion, the flow chart. There's some examples here. These are the 2 most common. They're not the only way to go, but I would suggest if you're going to deviate from this, maybe ask me about it, make sure it's not something that isn't going to be acceptable. So you start with your group and then you talk about each test you did in chronological order of when you did it. And so you name that test and then you list every single species by name that you eliminated. And for if you're in the Enterobacteriaceae or the Bacillus, it's a lot of species. And so the modules do have species lists that you can use, but it's really up to you to make sure that the accurate eliminations are being portrayed. A couple of things to know about how the format of saying these names, writing them down, they must be italicized, the genus needs to be capitalized, and the species needs to be lowercase. So if this was, say, an organism called Salmonella blah, okay, then you could either write out Salmonella, capital S, Salmonella, lowercase BLA, or you would have s.blah, that's also acceptable. So just like Escherichia coli or E. coli, both acceptable, but they have to be italicized. The E of E. coli would be capitalized. The coli part would not be. So if you're not sure how to say scientific names at this point, make sure you ask me, because I will take points off for that. Then you go through to the next test and you say what was eliminated. Be honest. If you hit a test that you did and it doesn't eliminate anything, you would just say no eliminations. Don't try to hide that from me. I will find out. And I will, there's a small deduction for having a test that you didn't need to do where it doesn't eliminate anything, but there's a much bigger deduction for being inaccurate in this process and for trying to mislead me when you write your lab report. But again, if you order a test that does not exist for your species, you don't have to include that. And for the flow chart, you don't have to tell me that something was inconclusive, and the inconclusives do not count against you. So let's suppose that you ordered a 10% salt test and what the technician said was inconclusive. That shows up in your results, but it doesn't show up here in your flow chart, and it doesn't count against you as a test. There's no way you could have known, right, that that test was not going to give you some information. Jade, go ahead. What's your question?
So if, let's say we ordered a test because based off of the data, it looked like we could possibly eliminate a good amount of species, but then how it came out, it turns out we weren't able to from that test. Is that the kind of thing that we lose points on?
It depends on the exact details. So you can always predict this because, if you're ordering a test and you look at what possibly could come out of that test, you'll have to see there's some positives, there's some negatives. Maybe there's also some inconclusives, but there has to be at least some positives and negatives so that when you get the result back, unless it's inconclusive, you're definitely going to make an elimination of some kind. If it's inconclusive, that's not your fault. The deductions are small. And so it's only if you make a ton of mistakes that it's going to have a significant impact on your grade. It sounds like this is a situation that might cost you a point. That's what it sounds like. But without looking at it more carefully, I can't be sure that I'm completely understanding the situation. Does that sort of start to answer your question for right now?
Yeah, that helps. And if we I'm not sure when these will be done being graded, but is it possible to have a meeting with you to go over points that were lost on it?
Yeah, of course.
After the fact?
Yeah, the only thing that I'll just warn you about, just so that everyone understands the situation, this thing takes me ages. I take a long time to grade these, and I don't promise they're going to be graded until like the last minute, and basically grades are due. Now, with that said, you are always welcome to have a meeting with me once grades are released. Just say, I want to meet with you to look over this so that I can have some peace of mind and also make sure that I, that everything was made sense to me. There's nothing that I, that was mistakenly graded down or whatever. Happy to do that for anybody that needs that. But yes, as to when they'll be graded, it will probably take me the entire week after they're submitted and then some to get everybody's done. And I don't release the scores until everything is graded. Or at least until the deadline has passed, and if there's extensions because of medical emergencies and stuff like that. So it could be a while before you know for sure.
Okay, that's fine.
And that's not ideal, and I totally get that because this is a high stakes assignment and you put a lot into it and you really want to have your answers sooner. And so that's just being real. Unfortunately, I wish I could be a little faster. But yes, once the scores are out, if you have any questions at all, please don't hesitate to ask, because I want you to have peace of mind around it and to understand it. And also, if I made a mistake, then I'm happy to correct it.
Okay, thank you so much.
You are welcome. Okay, any other questions? It looks like Jessica has a question as well. Go ahead.
So after listening to Jade, now I'm kind of freaking out, but
it's okay. Take a breath. It'll be okay.
Well, yeah, because when I'm listening to it, I'm like, oh shit, great. Because I had 2 of those kind of tests. But the thing is, when I got those results back, it was like the species that were like candidates were either that, say my test came back positive, right?
Mm-hmm.
and I, go to my table, and the ones that I are still candidates are either positive, but they're inconclusive. So am I going to lose a point for that?
How can they be both positive and inconclusive? I guess I don't understand.
Maybe I'll have to email you or talk to you about it after.
Are you Enero?
Am I what?
Enterobacteriaceae?
Yes.
And so when you say positive, you mean like 89?
Or 90. I was going off of 90.
Yeah. Okay. So how can I see? Okay.
Oh,
I see what you're saying. I misunderstood you. I understand what you're saying. So what you're saying is that you got a test result back,
yes.
and the species that remained
Yep.
were either positive or inconclusive, and your result was positive, so it didn't allow you to eliminate anything.
Right.
And the simple answer is yes, that might cause you to lose a small point because you should have known not to order that test if that was what was going to happen. Now, with that said, it a little bit depends on where it happens in the process, right?
Mm-hmm.
So early in the process, when you have tons of species and you're ordering like multiple tests at once,
Mm-hmm.
there's a little more leeway because, you might order 4 tests as long as when you ordered those 4 tests, you really had 40 or more species. It wasn't that you ordered 4 tests and you had 10 and you were just freaking out.
Right.
Right. But if you had legitimately like 40 or more species and you ordered 4 tests and one of them, by the time you process it, like, that's less likely to cause a problem. It's later when you've got a small number of candidates. What I do expect is that people will not just be throwing shit at the wall, right? You
Right.
have 10 things and you're not just like, let me just throw like 4 tests at 2, even though that's not
Right.
what's supposed to happen, and without really thinking ahead. So I need to see your strategy.
Okay.
And and it might or might not be a problem depending on where in the process it happens. Right.
Okay.
Yeah.
Yep.
Does that make sense? And maybe that
Yes.
helps to answer Jade's question as well.
Yep.
So the early steps are
Mhm.
ones that I don't pay as much attention to because I know I've given you a little bit of leeway. Like you can order 4 tests or 3 tests. But if it's like you're down to like 17 species
Mhm.
and you ordered a bunch of tests and you just over ordered, you
Right.
weren't strategic enough, you weren't patient enough, then that could cause you a deduction. Yes.
Okay.
Does that help?
Yep. That helps. Perfect. Thank you.
All right, Julia.
Yeah, mine's gonna be very quick. So I ordered the INVIC test, but the method read was inconclusive. So I just left it out and then I just keep counting. And then at the end, the method test came back, correction, when I had 3 species left. So at that point, because of the correction of method read test, I eliminated just 1. So I'm going, no problem. So when I put the flow chart, I'm going to put my method read test almost at the end.
Yes.
Is that how I do, right?
Yes.
Just the way I received,
Yes.
right?
Yes.
Okay.
Yeah.
Thank you.
And you might want to make a note about that in your
Mhm.
lab notebook. You wouldn't
Yes, I
put
did.
it in
Oh.
the
Star, what?
you wouldn't put it in the lab report because you don't want to put things into the lab report that are not sort of the science. There, you don't pull the curtain back on this movie, right? You're not going to say in your lab report, you won't say things like the technician screwed up and then I got the
Mhm.
second result. If you want to do it kind of like that, you could say something like the methyl red test was inconclusive in your results table. In the how you could document the most easily is you might have methyl red inconclusive with a date early in your results table and then later on a methyl red test again. And if I see that, I'll be like, oh, I wonder what happened. I'll go read your lab notebook. In the lab notebook, it'll be properly documented. Oh, the technician sent this correction. There was a mistake made early on. And so
Okay.
then that's not your fault.
Okay,
Yeah.
so I'm gonna put method read in order, but the first method read will be just inconclusive, and then at the end, I'm gonna put method read test again, and then I'm gonna put my
In the results, it could appear twice, right? Because you put in the results table, you
A
put both
result. Okay,
inconclusives
okay.
and observations of real data.
How about
But in the flow chart,
Yeah.
it should only appear at the end.
at the end?
Yeah.
Yes.
No, and that's fine. Even if you did that on accident, like let's suppose that you forgot to order your methyl red test. It's like, oh, well, you just did a different version. I recommend the INVIC series because I know it'll help you. But if you want to be like, John Wayne in the Wild Wild West and just ride into the sunset on your own in the Enterobacteriaceae, that's not wrong.
No, I didn't do it. I didn't.
I know that, but I'm just
Okay,
saying,
okay.
you know, for that reason, I wouldn't penalize you for putting it later. Yeah.
Okay. Well, I couldn't help it because I had to show the eliminated
Yes.
species, but then I can't.
Yeah.
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 Okay, thank you.
Yeah, no problem. Okay, it looks like Jessica, go ahead.
All right, so I'm gonna ask kind of a follow-up question on everyone else's. Um, I also have one of those that didn't eliminate anything, but it is one. It's kind of a silly one, and I know now that I probably shouldn't have ordered it, but it was a group of 4 because it had so many species. Do you want me to write in the species eliminated table that it was a group of 4, or are you just gonna refer back to the lab notebook?
I look at your lab notebook. Yeah.
Okay. And another thing is, since I already did that table, is it okay if I leave the inconclusive results that they eliminated no species?
Oh, you mean for the flow chart?
Yeah, for the flow chart.
Take
Because
them out.
I just included everything. Okay.
Yeah, take them out of the flow chart. They can go in the results table, and they should go in the results table, partly because you might need them later, right? At the very end, you might need them, and you might not, but some people do. And so that's why they need to be in the results table, especially if you ended up needing them to help like sort out between 2 or 3 or 4 species at the very end. But they don't belong in the flow chart because you didn't actually use them to eliminate. Sorry, my cat keeps walking in front of the camera.
Okay.
You didn't use them to eliminate anything unless you did it at the very end. So it doesn't belong in the flow chart. And it'll just be an accidental distractor. Like, what if I'm tired at 2 o'clock in the morning and I'm like, oh, no species eliminated, down for you. You know, you don't want that. So I would take them out.
Okay.
Yeah. Does that make sense?
It does. I'll just leave the one that I used at the very end to eliminate the rest.
Yeah.
Okay. Perfect. Thank you.
And looks like Roman.
Hi again. So, a bunch of people are asking. So I think I thought it was clear, but apparently it's a morning or something. So I have inconclusive test or the no data available test, in other words. So does that only go in the results table, or does that go anywhere else? I'm sorry, I just confused me.
And those are actually, I don't know if you meant to say it this way, but what you actually said are 2 different things. So I'll answer for both things. So you might have, and I'm not saying you did, it's not you, many people did not, but it's possible to have accidentally requested a test that does not exist for your group. And you would know that if when you asked for it, the technician said this test is not available. This test is
yeah, that's what I got.
Means you shouldn't have ordered it at all because it's not even possible to get it. So that goes in your lab notebook. Everything goes in your lab notebook, but that test not available doesn't go anywhere in your lab report because it's not actual data of any kind, right? It just, there's no results at all. If on the other hand, the technician wrote back, data is inconclusive or test result is inconclusive, what that means is this is a test is available, you could have run it, but for your particular species, the answer isn't very clear. Right? And so that goes into your results table.
Okay.
Okay. But it doesn't go into the flow chart unless you actually used it at the very end to eliminate something. So because remember, when you get an inconclusive result, you file it away in your lab report, in your lab notebook, and you don't use it to eliminate anything. So it shouldn't show up in your flow chart unless at the very end you were like, I have 4 species. Now I've got to figure out which one is the most likely. And then you would sort of summon up all of the powers of data. Like you would think about all of the data in total, including any inconclusive results to try to make that call. Does that make sense?
Yes.
Yeah. Good question. So does that like clarify it though for you? Like, do you feel like, yeah, now I understand?
Yes.
Okay, good. Miss Jade, tell me what's on your mind.
Sorry to have so many.
Yay.
So I was just wondering if this needs to be included in the lab report itself. So I ended up with 2 remaining species that I could not run any more tests for, and I ended up eliminating them just like you said. It is in my journal of that, how I did that process. Do I also need to include that process in the lab report somewhere?
Yes. And so it's going to go in at least in the body of the text, all right, when you're talking about you do your little, how did I figure out my group? And then you have your paragraph about how you figured out your species. Then at that point, you would say, after all these tests were performed, the remaining candidates were blah and blah. However, yada yada species is much more likely for reasons X, Y, and Z. These are the matches and mismatches. If you want to add that into your flowchart, which is a great idea, you can have your final line item being whatever particular tests you looked at that helped you to distinguish between them. And then you would say, this guy was eliminated by that, or was less likely, and this is the one that seems most likely. So there'd be sort of a final node to your flowchart, if that makes sense.
Okay, so put it in the flow chart and then also say something about it after you talk about the test that you ran.
Yeah.
Okay. Thank you.
Awesome. Okay. Let's see if any chat things. Okay. Any other questions? Okay. Let me just go back here. We'll resume our wander through this lab report and see what pops up for you guys. So far, it's been, oh dear, what happened? There we go. I think helpful to sort of surface the issues and the questions. So here's your flow chart. You walk through it, you make your eliminations, make sure your species are all there. This is the question that Julia brought up earlier. My intention was to have you write a series of paragraphs about your elimination process that integrates the structural and metabolic features, but this is not very clear, and some of you may have already started writing. And so I will edit this, but what's required is the first paragraph has to be, how did you get to your group? Because that's not required to be part of your flowchart. So you have to say the unknown number 46 was gram-negative rods, and the thioglycolate test showed that it was a obligate aerobe, and the oxidase test was positive. These, whatever they tell us, you interpret that information. This tells us that the organism can only do aerobic respiration and that it has cytochrome C oxidase as part of that metabolic process. These two tests revealed that this organism is Pseudomonad or whatever it is. And then that's your first paragraph. How did you get to your group? After that, you can either run through the eliminations in text, talking about each test in turn and what it was, positive or negative, what it tells you about the organism and what it eliminated, summarizing number of species. Or you can just follow these instructions where you just put all of the information about the structural and metabolic features into one paragraph. However, if you didn't come to just one species at the end of your flowchart, which is what Jade's question was, and this is true for probably like 25% of the people, then you do need an additional paragraph that says, at the end of all these tests, there were 3 species left, so and so and so and so and so and so. In order to distinguish them, the entire data set was looked at. And, the most likely species is E. coli because it was a perfect match to all the data, whereas Citrobacter, whatever, was inconclusive for several tests that were positive or negative. And, Pantoa agglomerans was positive when the unknown was inconclusive. So you give me your rationale. If you had to pick and do a best fit, you give me your best fit argument. All right. A few things about this, and this was mentioned earlier, but I'll just say it now. You don't want to restate observations. Observations go into the results section. So there were bubbles in the catalase test, or the fermentation test was a red broth with no bubble in the Durham tube. Those observations do not belong here in the discussion. So instead you would say the thioglycolate test revealed this, that it's a facultative anaerobe, suggesting that it's capable of growing in both aerobic and anaerobic conditions and does aerobic respiration when it's possible. Or the catalase test was positive, indicating this organism has the catalase enzyme and can detoxify hydrogen peroxide. So you don't want to say that you saw bubbles or that you saw pink cells or whatever. This part here is optional, and if you did it, it's fine. But if you don't do it, it's also fine. Because what happened was, for some reason this quarter, I don't know, maybe it's just been a harder quarter for many people, but a lot of people in the class are a little bit further behind than normal. And so adding this in makes it a bit of infeasible and stressful at the end of the quarter to really get everything done. Some people are still working on their ID and they're not finished. So that's why I've created this to be extra credit for those of you who got there and did a due diligence and were making good progress and got here. Good, you can get extra credit for having done this, but the answer to this is going to go into that separate Dropbox that you guys probably saw. And if you put it into your lab report, no problem, right? But it's not going to get graded. So you can say whatever you want. You can put, my cat has cute feet in here, and I'll probably notice because I read everything, but maybe don't, you know. But anyway, all I'm saying is it's fine. Okay, so make sure that it's somewhere in this section, it is extremely clear what you're identifying, what you have identified as your species. Don't make me hunt for it. Make sure it's quite clear and that it's not hidden, or that you assume that, you know, I can see it, and make sure it's written in the text somewhere. And that's probably a good place is to do that right at the start of this section, which is the research section. The research section needs to be substantial. Your whole report is single space. So 3/4 of a page is not like, you know, 3 sentences. And you are researching what is known about this species. You know, what is sort of most relevant and important and topical today about this species. You are welcome to include information that you find in the Bergies, information that you find in interesting journal articles, information you find in the news. All of this stuff is perfectly fine, but do make sure that you kind of cover what the most important things are that I would know about the species, so that I know you did a good job of looking it up and you didn't just go Google on Tuesday at 2 a.m. You got 3 things and then you're done. Okay. The references and the in-text citations I will also cover again, just for those of you who are listening at home on the recording. All the references in this list must be ones you actually used to support information you actually said in this section. Okay, so you don't cite the lab manual. The lab manual, you don't cite it. First of all, you didn't do any of the work actually, right? So it's sort of, and we don't talk about the procedures, but it's also not a peer-reviewed document with any useful information about the research of the organism. So this reference section is for this research section. Make sure your references are complete so that if I use them, I can find exactly what you were looking at. So if it's a URL, it's the entire URL. Make sure that if it's an online source, it's not a wiki, it's not a blog, and most important, because people forget this part, it's not an edu, which indicates that some teacher who's like probably smart and super nice wrote it, but nobody looked it over, and so they might have made a mistake. In the in-text citation, every one of these references is going to have at least one in-text citation. Somewhere in here, there's information that you used which justified including that reference here. And that in-text citation should be in brackets, something like authors et al. Date, or as Roman asked, what if there's no author because it's on like the CDC or the World Health Organization or something like that, some government, NIH perhaps, in which case you give it a short abbreviated title that has a unique tie to whatever the thing is listed here, and so that I can easily find it and distinguish it from the other things and look it up and make sure that you said things that were true and that you sourced them appropriately. What's the other thing? I think that's pretty much everything. All right, having said all of that, again, I'll just say again, single spaced, please. Make sure it is a PDF or a Word document so that I can actually read it. As Jade asked earlier, the lab notebook does go in the Dropbox. It goes separately, so don't attach it directly to your lab report. And with that, are there any final questions? Go ahead, Jessica.
All right. Sorry, this is probably a repetitive question, but
Absolutely fine. If it's repetitive for you, someone else is also still wondering about it, and they appreciate you asking.
so now I'm kind of seeing how strict this I feel like this is going to be great and I'm kind of freaking out. Um, so the extra credit, I noticed that a lot of them are out of like 5 instead of 0. So I don't know if they'd be considered extra
So
credit.
you're wondering about how is extra credit graded, or how does it affect your grade at the end of the story? And right now, all the extra credit is actually not impacting your grade at all, even though it shows out of 5 or out of 0 or whatever it's set to at this particular moment in time. It's in a category on Canvas where it doesn't contribute to your grade. It's not contributing to your grade right now.
Oh, okay.
So what happens is at the end of the quarter, I export everything into an Excel file and I do your grade by hand. Every once in a great while, Canvas has done something wackadoodle that I don't like. And so I do the grades by hand. I double check everything. And then I compare to make sure that it's like at least as high as Canvas, but the extra credit should boost it a little bit. So the extra credit is done completely by hand. I add up your points. I think it's like whatever the points total in the class, which is somewhere around 720 or 750. Each extra credit point is 1/720th or 1/750th of a percentage. So, sorry, of a point, which is then times 100. So whatever that is, let me do the math real quick so you kind of have a sense of what I mean. So each extra credit point is going to boost your grade by something like 0.125%.
Okay.
So like, let's suppose you got 10 extra credit points, theoretically. Then you would maybe get an extra 1 or 1.2% boost to your grade at the end of the quarter. Does that sort of help? It's not a ton. What it is like, let's suppose you're right at a grade point, right before one where you might be boosted up. And so this could push you into that next level.
Which will definitely probably help for a lot of us. So if I remember right, there's 4 extra credit opportunities, one from Post Lab 3, the Zoom check-in, ID check 4, and then the ID questions, right?
Yeah, if you're in the online only sections, yes, there's the check-in one. The people listening at home, if you're in the live stream, there was not a extra credit meeting option. And if you're feeling sad about that, just know that typically the average in the live stream is a little bit higher anyway. So yes, that's what those are the extra credit options. There's a final evaluation of an informal evaluation at the end of the quarter, and you can get extra credit by filling that out. There's the mandatory, or not mandatory, the extra credit check-in meeting, and the step 4, and then the post lab 3. Yep.
Okay. Thank you so much. I have one last question.
Go ahead.
So you said test 2 is the hardest, right? How's the final compared to test 2?
Oh, great question.
Is it like questions we've seen, or is it different?
The concepts cover everything that would have been on previous tests. So there's not going to be like concepts that are reintroduced that you were not tested on. However, the questions are not the same. I reword them. I ask different ones. I combine, for like there's not a ton of those like really hard ones at the end with lots of drop downs, but for the logical reasoning ones, it's going to be some different version of what you've seen before. So knowing how you did on the previous exams is helpful, but it's not like any of the questions show up identical, with the exception of the true falses. Sometimes I'm just like, I really like this true false question, I'm just going to reuse it. Does that help?
It does, because I know that test 3 will probably be a lot easier, but the final I'm worried about, because
I'm sorry, and thank you for asking that again, because I didn't quite give you the full answer that you asked. The final typically is kind of similar to exam 1.
Okay.
Everyone's different, right? If you just do a really good job of prepping, maybe you're not going to be typical, but typical exam 1 and the final are kind of on par. Exam 2 is usually a little bit lower. Exam 3 is usually a little bit higher. You'll notice that I did a little bit of a curve on exam 2, so it's a little bit closer to exam 1 than it was before, but still low. So exam one is often a pretty good indicator of how you'll do on the final, but it really just depends like how you prep for it, and you can certainly beat those odds. And the other thing to know about the final is that 50% of the time it helps you. That's just the average over many quarters. On average, if you take the final, half the time it boosts your score a little bit. Like that means that test is then higher than a previous test, usually exam 2, but it's not always exam 2. So, I mean, obviously we don't have a crystal ball, but you can probably expect if you do a good job of preparing for the final, you have a decent chance of actually having that help you.
Okay, perfect. Thank you so much.
You are welcome. Awesome. Okay, well, those were excellent questions. Please, after you've had a chance to sort of digest this information, if you have lingering concerns or questions, you should feel free to email me. And for those of you in my live stream class, I will see you tomorrow.